any syllabus. Then apart from the academic support, which is a common norm for all the institutions, here at Oracle, what makes it different and sets it apart from the other institution is that you have personalized counseling sessions. So it is like a very personal therapy session where from the apart from the academic support, you can get all that extra guidance and support from our end, which is also uh, like not related only to your academics, but non-academic support as well. Then we have 3 different subscription plans at Oracle. According to your need, you can available any of the 3 plans. We have plan which is for 3 months starting from 15,000. Then you have the standard which is for 6 months and it will cost you 30,000, and the premium plan which is for 50,000 and it comes annually. So in the premium plan, you also have live doubt clarification sessions and one-to-one personalized guidance. Then apart from that, you can get an extra 20% off if you use my promo code as displayed here on the screen. Then talking about the internal exam marathon series, this series has been specifically curated to target your university exams, which are approaching in a while. So this is a rapid revision series of all important topics. We are trying to cover the most frequently asked and the important topics from the examination point of view. Then volatile quick facts will also be covered. Multiple choice questions will also be covered so that your top grades can be assured in the exams. Okay. So beginning with the chemistry of amino acids. So your amino acids, they are the building blocks of proteins. Just like when you want to build a building or an apartment, you use bricks. Similarly, if you want to build a protein, make a protein, you will be needing amino acids. Okay, so by joining amino acids, you make a polymer, which is known as proteins. And then different proteins, they are, the primary structure is formed, then they are forming the tertiary, the secondary, quaternary structures, and ultimately you have all the functional proteins in your body. But where does it all begin from? It begins from the basic unit, the building blocks, which is amino acids. So today we'll be talking about these amino acids, their chemistry, their classification, their properties, and everything. Okay. So talking about the basic structure of an amino acid, you have this carbon here, which is known as the alpha carbon. To this alpha carbon, you have this carbonyl group attached, and you have another group which is known as the amino group attached. Okay, so you have this carboxylic acid group, and you have this amino group, and you have this hydrogen atom. So three different groups, these 3 different groups that are attached to this carbon, they are common or constant for all the amino acids. Okay, so this much of it is common for all the amino acids. Now what is different is this R. Okay, this is the variable side chain. So this is different in different amino acids. So that is what sets different amino acids apart from each other. That is this variable side chain, this group R. Now most amino acids are alpha amino acids. Okay, that is to the same carbon you have your carboxyl group attached and your amino group attached. Okay. Now, apart from that, you have non-alpha amino acids like beta-alanine, beta-amino isobutyrate, and gamma-amino isobutyrate. So, what is the difference between alpha and non-alpha amino acids? That is, your carbonyl and amino groups are not attached to the same alpha carbon. Okay. For example, I'll show you this with the help of the structure of beta-alanine. Okay. Although you need not know the structure, but for the sake of understanding, I'll make it. So this is your functional group carbonyl. Okay, so the carbon next to the functional group is your alpha carbon. Okay, now you see to this alpha carbon, instead of the amino group being attached, there's another carbon, beta carbon, okay, and to this beta carbon your amino group is attached, okay. So this is the structure of beta alanine. So beta alanine, beta amino isobutyrate, gamma amino isobutyrate, these are examples of non-alpha amino acid. It becomes important from MCQ point of view, like there might be like three options might be of alpha amino acids, and this one there might be like one option like beta alanine or gamma amino isobutyrate, and the question might ask you which of the following is a non-alpha amino acid, okay. So you need to know the examples. Okay, now moving on to the peptide bond formation. Okay, so when I say in order to build proteins, you have to join amino acids together. So how do you join them? When you're building an apartment, you're putting bricks together. How do you join the bricks? By cementing them. Okay, so here that cement is your peptide bond. You have to join amino acids together in order to form a polymer by a peptide bond. So your peptide bonds are joining the amino acids together. Okay, now how is it formed? The carbonyl group of one amino acid and the amino group of the other amino acids, they are engaged in a peptide linkage. Let's understand this with the help of an example. You can see here, yeah. So you see this is the carbonyl group of one amino acid and this is the amino group of the other amino acid. So they are engaged in a peptide linkage. How? By the removal of water. So a water molecule is released. You can see OH from here and H from here. So this forms H2O. This water molecule is released and a peptide bond. You can see a peptide bond is formed between what? The carbonyl of one amino acid and the amino group of the other amino acid. So now your carbonyl and amino groups, they are engaged in a peptide linkage. Now what is free? You R, this variable side chain. Now this R is the only free group, okay? Is the only free group. Okay, so this is the only free group that is left. So this variable side chain is determines the properties of the proteins. So this variable side chain R, it determines the properties of proteins. Because your functional groups, they are engaged in a peptide linkage. So the only free group now you have is your variable side chain. So the properties of the protein are determined by the variable side chain. And this peptide bond that is formed, it can only form hydrogen bonds, okay? And it is not involved in any other reaction. It only forms hydrogen bonds. It is not involved in any other reaction. So the classification of your amino acids, it is based on this side chain, okay, the side chain characteristics. So your amino acids are classified based on the side chain characteristics and other categories. So that was about the structure in the peptide bond. Now moving on to the classification of amino acids. So your amino acids, as I said, they're classified based on side chains, based on the characteristics of side chains, on their metabolic rates and their nutritional requirement. Okay. So based on side chain characteristics, you have aliphatic amino acids. So your aliphatic amino acids, they may be simple or branched. In simple, you have glycine and alanine. And in branched aliphatic amino acids, you have leucine, isoleucine, and valine. So branched amino acids, you can remember by a mnemonic as LIV. LIV live. So L stands for leucine, I stands for isoleucine, and V stands for valine. Okay. So your aliphatic amino acids, they are straight hydrocarbon chains. Now, the simplest amino acid is your glycine. Okay, I'll show you the structure of glycine. This is your alpha carbon. To this, you have the carbonyl group, the amino group, and then you have the H. So this is like the constant in every amino acid. Now you have the variable part, and in glycine, this is simply a hydrogen. This is your glycine, the simplest amino acid. I'll just zoom in it a little. Can you see now? Yeah. Okay, now drawing the structure of alanine. To draw the structure of alanine, again you have the alpha carbon. Then to this you have the functional groups, and then you have H. So this is the constant part for every amino acid, and then you have in the side chain, you have a methyl group. In glycine, in the side chain, you have a hydrogen, and in alanine, you have a methyl. Okay, so these are your glycine and alanine. Now moving on to hydroxyl containing amino acid, you have your serine, threonine, and tyrosine, where tyrosine is aromatic. Okay, we will study about tyrosine in detail when we take the metabolism of tyrosine. But for now, just know that it is aromatic. So these are your hydroxyl group, OH containing amino acids. So your serine. If we talk about the structure of serine, you have the common part. Okay, and what is different in serine is that it has hydroxyl group. Okay, so you have CH2OH hydroxyl group. So this is your serine. Now, often while studying the key enzymes of pathways, you have come across phosphorylation, dephosphorylation, glycosylation, all that stuff. So here your hydroxyl amino acids come into role. That is, these hydroxyl containing amino acids, they are site for phosphorylation, dephosphorylation, glycosylation and all. Okay. Okay. Only for the sake of explaining, I'm drawing the structures, but you it is very much okay if you do not know the structures. Okay. Now moving on to acidic amino acid. These acidic amino acids, they have an extra COH group in their side chain, and they include your aspartic acid and your glutamic acid. Okay. So they have extra COH in side chain. So if we talk about the structure of aspartic and glutamic acid, so you have the common part, which is common to all the amino acids. I'll draw it quickly twice. Okay. Now let's see the variable part of both of them. So in aspartic acid, in the variable part, you have CH2COH in the side chain. So this forms your aspartic acid. And for this amino acid, its corresponding keto acid is oxaloacetate. And for your glutamic acid, in the side chain, you have CH2, CH2, COOH. So this is your glutamic acid. And the keto acid for this is alpha ketoglutarate. So the only difference between aspartic and your glutamic acid is of one carbon. Okay, your glutamic acid has one carbon more than the aspartic acid in the side chain. So that was about glutamic and aspartic acid, and also when they are ionized. Okay, so they are known as aspartate or glutamate. Okay, so this aspartic acid and glutamic acid that I've drawn, they are in unionized state, but when they are ionized, they're known as aspartate or glutamate. So when ionized, known as aspartate and glutamate. Okay. If we talk about the structure, I'll just show you the example of aspartate. Okay, glutamate, it is exactly the same. So this is your aspartate. When ionized, it is known as aspartate. Okay. Now talking about amide containing amino acid. Okay, so amide containing amino acid, they have the group CONH2, and they are examples are asparagine and glutamine. We talk about the structures of asparagine and glutamine. You have the common structure, which is same, that is NH2CCOH. Let me draw the variable part. So the variable part is where you have CH2CONH2 group. So this becomes your asparagine. This. Okay, similarly, you have glutamine. In glutamine, you have exactly one carbon more, just like aspartic acid and glutamic acid. Okay. So if I draw the structure of glutamine, it will be something like this. You have carbon, this is the constant part, and if I draw the variable part, CONH2. So this becomes your glutamine. Okay. Talking about sulfur containing amino acids, that is, they have sulfur in their side chain. Example includes your cysteine and methionine. Now this is important, knowing the structures of cysteine and methionine. I'll tell you why. Because in the MCQs, first let me give you the structures for both of them. So if we talk about cysteine first, I hope it's not too small, you can see it's visible. Zoom. Okay. So talking about the structures of cysteine and methionine. So this is the constant part. Now let me draw the variable part for cysteine. Okay. Now this is your cysteine. And this is your methionine. Now, do you see any difference in the two of them? Your cysteine, it has a free terminal sulfhydryl group here. So it has a free terminal sulfhydryl group, whereas in methionine, your sulfur, it is trapped between two carbon. It is engaged in a thioether linkage. Okay, it is engaged in a thioether linkage. This is a thioether linkage. And whereas in cysteine, it has sulfhydryl group, which is free and terminal. Okay. Use carbon, oxygen, carbon. This is your ether linkage. But when your oxygen is replaced by sulfur, this becomes your thioether linkage. Okay. So in methionine, you have a thioether linkage, whereas in cysteine, you have a free and terminal sulfhydryl group. Now this thiol, also known as thiol, this is providing it polarity. Okay. So your cysteine, it will be polar because of the free terminal sulfhydryl group. Now the question, an application question is about lead sulfide test. Okay. If we talk about sulfide, lead sulfide test. So you get an MCQ which says that your sulfur compounds, they give lead sulfide test, and both your cysteine and methionine, they are sulfur containing amino acids. But why is it so that your Methionine does not give lead sulfide test, whereas it is answered by cysteine. So it is very clear now, because your cysteine, it has a free and terminal sulfhydryl group. So your sulfur will be liberated and given to lead to form the black colored lead sulfide. Whereas in methionine, it is trapped between two carbons. It is engaged in a thioether linkage. So it will not be liberated and given to lead to form lead sulfide. So that is the application based on this. So I'll write this down for you so that when you're revising, these notes will be sent to you. So when you're revising, you know it, okay? So in cysteine, your sulfhydryl group is liberated, whereas in methionine, it is not, okay? It is, so cysteine gives this given to lead to form lead sulfide. So the black color is formed, whereas in methionine, it is not. Now talking about basic amino acids. So basic amino acids are your histidine, arginine, and lysine. We'll talk about them when we take their metabolism. Your basic amino acids, they have extra amino group. Extra amino group in their side chain. So lysine, it has epsilon amino group. Your histidine, it has an imidazole ring. And your arginine, it has a guanidinium ring. Now moving on to aromatic amino acids. You have phenylalanine, tyrosine, histidine, and tryptophan. We'll study about the metabolism. That is when we will take the structures. Your phenylalanine, it is, we've just seen the structure of alanine. Okay? So in alanine, there is a methyl group here. But when it becomes phenylalanine, you have a phenyl ring attached. So that is the reason why it is known as phenylalanine, because to your alanine, a phenyl ring has been attached. This was alanine. Okay, so this becomes phenylalanine. Now moving on to the next classification of amino acids, which is based on the side chain characteristics. So according to the characteristics of the side chain, you have polar and non-polar amino acids. Your polar amino acids, they again are categorized into charged and uncharged. So those amino acids which are uncharged at physiological pH, the side chain is not ionized. They are your polar amino acids. Okay. Now your polar amino acids, if they're charged, they include your basic and your acidic amino acids. Now those polar amino acids which at the physiological pH, they do not carry any charge, and they are known as uncharged polar amino acids, and they are your hydroxyl containing, like you have your serine, you have threonine, and then your glycine is also included among the polars, but it is the least polar among polar amino acids. Okay, so you have glycine as well, you can write. Then you have your amides. Okay, that is your glutamine and asparagine, and then you have cysteine. We all know why methionine is not included, because its sulfur is not providing any polarity, because it is engaged in thioether linkage. Okay, so your uncharged polar amino acids, when their side chain is not ionized at physiological pH, they are your uncharged, and your polar charged amino acids are your basic and your acidic amino acids. And non-polar amino acids are the ones which do not carry any charge. So rest of the amino acids which have not been included in this list, they automatically fall into the non-polar category. Now based on metabolic state, they are categorized into glucogenic, ketogenic, or both. Both includes that is they are both ketogenic and glucogenic. So they are both ketogenic and glucogenic. So your principal glucogenic amino acid is your alanine. And your ketogenic amino acids, your example are leucine and lysine. And for the example of amino acids which are both ketogenic and glucogenic, it is phenylalanine. Then you have your isoleucine, then your tryptophan, and your tyrosine. Okay, so those amino acids which can be converted to glucose, they are glucogenic amino acids, and those which are converted to ketone bodies, they are ketogenic amino acids. Now, based on the nutritional requirement, they can be essential and non-essential. Essential are those amino acids which are supplemented through diet. Supplemented through diet. And non-essential, they are synthesized in the body. Although truly speaking, technically your essential amino acids, they can also be synthesized in the body. But the pathway for their synthesis, it is very long, more than 7 steps, so 7 different enzymes are needed. So to conserve ATP or energy, the body is rather preferring to take them through diet. Now moving on to the general reactions or general amino acid metabolism, that is the reactions of amino acid in general. So they undergo 3 different general reactions, that is decarboxylation, deamination, and transamination, out of which transamination is very important. You often get this for short notes. So talking about decarboxylation and deamination. So decarboxylation, the name itself tells you that you are removing carbon dioxide. So in decarboxylation, after removing carbon dioxide, what do you end up with? You end up with amines which have very important biological significance. Okay. So let's understand this with the help of an example. So this is your example. You can see this is your alpha carbon. To this alpha carbon, you can see a COH group is attached and an amino group is attached. Now what is happening? Decarboxylation is taking place. You see this CO2 here is removed. The one shown in purple, I've highlighted it. So decarboxylation is taking place. Decarboxylation. That is removal of carbon dioxide. Now what do you get? You get this amine. So amines which are formed on decarboxylation of amino acids, they have biological importance, okay? And you can see this carbon dioxide has been removed. Another very important point, that PLP is the coenzyme for all amino acid decarboxylations, okay? Now talking about deamination. So deamination of amino acid, it results in the formation of keto acids. Let's understand this with the help of this example. So you can see alanine here, this is amino acid. You are removing this amino group. You can see ammonia here, it has been removed. And what is formed? You can see a keto group has been formed here. So you're ending with a keto acid, alpha keto acid. So the keto acid for alanine is your pyruvic acid. Okay. Now let's talk about transamination. Very important. This. So what is transamination? It is the transfer of amino group from one amino acid to alpha keto acid to form a new pair of amino acid and keto acid. So understanding this with this diagram, you have this amino acid, okay? This is your first amino acid and this is your first keto acid. So you already have a pair of amino acid and a keto acid. Now what it is saying? It is saying this transfer of amino group from one amino acid to the alpha keto acid. Okay, so from this amino acid, which I've labeled as 1, from this alpha amino acid, the amino group is being transferred to this keto acid. Here in this case, this keto acid, we've taken the example of alpha ketoglutarate, okay? Now it is being transferred to this keto acid, which here is alpha ketoglutarate. So obviously, after accepting an amino group, this keto acid will form its corresponding amino acid, which is glutamate. And this alpha amino acid, after giving its amino group, it will form its corresponding keto acid. So you end up with a new pair of amino acid and keto acid. This is what is known as transamination reaction. Okay. So there is transfer of amino group from one amino acid to the alpha keto acid to form a new pair of amino acid and keto acid. So what is happening? You can understand this with the help of a funnel. Okay. So it is something like there's a funnel here in your body. What is happening? Transamination is taking place, so all your amino acids is kind of passing to a funnel. Okay, that is that, and that funnel is your transamination reaction. So what is happening? You have any alpha amino acid. Okay, it can be any alpha amino acid, which after giving its amino group, it is changing into its corresponding keto acid. And what is happening, that amino group, it is taken almost all the time by alpha ketoglutarate. And depending upon the transamination, you have different enzymes for this transamination. Okay, so you have different transaminases like ALT, AST. So depending on the transaminases, you have them. What is happening now? It can be any alpha amino acid, which on giving its amino group, it becomes its corresponding keto acid, and most of the time it gives its amino group to alpha ketoglutarate. So when alpha ketoglutarate will accept an amino group, what is formed? Glutamate is formed. Okay. So in the body, actually, what is accumulating? Glutamate is accumulating. Okay. So the ammonia, which is toxic to the body, in all the organs, it is not released freely. Rather, it is concentrated as glutamate in the body via transamination reactions. So you see, the transamination reactions play a very important role in handling of this toxic ammonia. Okay. So the ammonia, which is very toxic. Okay. It is very, why is it important to handle this ammonia? Because it is very toxic. Okay. It has a lot of side effects. We'll talk about the toxicity of ammonia later. For now, just know that ammonia is very toxic. So it is very important that we regulate the levels of ammonia in our bodies. So this toxic ammonia which is released, it is not released as such in the body. Rather, it is accumulated. It's as glutamate in the body. Okay. It is concentrated as glutamate. And by which reaction? By transamination. Okay. So your transamination reactions are taking place in almost all the organs of the body. And what is happening? That ammonia which is being released, it is not released as such freely in the body since it is very toxic. Rather, via transamination reaction, it is being concentrated as glutamate in the body. Now let's talk about the general properties of transamination reaction. I hope transamination reaction is clear. It is nothing, it is very simple, just simply the transfer of amino group from an amino acid to another keto acid, but most of the time that keto acid is alpha ketoglutarate. So your alpha ketoglutarate, on accepting an amino group, it will form a corresponding amino acid, which is glutamate. So your glutamate is accumulating in the body. So ammonia in the body is rather accumulating in the form of glutamate. Now, site is mostly taking place in the cytoplasm, although certain transaminases are the enzyme which bring about transamination reaction. Okay, they are also found in mitochondria. Example, your AST. So the coenzyme is PLP. It is a reversible reaction. The ammonia is not released freely, it is concentrated as glutamate. I just explained how, because most of the time the other keto acid that is accepting ammonia is almost alpha ketoglutarate. Okay, so your alpha ketoglutarate is converted into glutamate. Now, the mechanism of this transamination reaction, it follows the ping pong mechanism, that is bye-bye reaction. Bye-bye reaction. Bye-bye reaction means the reaction is having two substrate and two products. Reaction is having two substrate and two products. Important, this ping pong reaction. Sometimes you might get asked about it. Now it is involved in the biosynthesis of non-essential amino acids. Okay, so your transamination reactions are involved in the biosynthesis of non-essential amino acids. So if we talk about what is ping pong, ping pong is your table tennis ball. Okay, so the mechanism looks something like this. This is your, consider this as a table. So what is happening? Your substrate 1, which is an alpha amino acid, it gives its amino group and it is converted into product 1. Okay, now what is happening? That, and what is that product 1? So alpha amino acid, after giving its amino group, will be converted to product 1, and that product 1, we all know it will be its corresponding keto acids. Okay, so it becomes a keto acid. So your substrate 1 has changed into the product. So one product has already been released. Okay, now what is happening? Your substrate 2, it will accept that amino group and will be changed into its corresponding amino acid. So this is what your transamination reaction. So you see, two substrates and two products are involved. So therefore it is also known as bye-bye reaction. And this looks like a table tennis ball. When you throw a ping pong or a table tennis ball on the table, this is how the movement looks something like this. Okay, like a ping pong ball. So therefore it is also known as ping pong mechanism or ping pong reactions. Now, so talking about the sources of ammonia. So alpha amino group of amino acids detoxified to glutamate by transamination. That is one source. Then you amino sugars, that is another source. Non-protein nitrogenous substances like nucleotides, phospholipids, porphyrins, and gut bacteria. So we're talking about that. Where is this ammonia in your body coming from? So first source is it from your alpha amino acids. Okay, that is the amino group of your alpha amino acids, which give their your amino acids, alpha amino acids giving their amino group to alpha ketoglutarate to form glutamate. Okay, so that is one source of ammonia in your body that is being accumulated as glutamate via transamination reaction. The other is your amino sugars. Remember, amino sugars, we spoken about them in GAGs. Okay, what are amino sugars? They're formed by replacing the hydroxylate carbon 2 of sugars with an amino group. Then you have other substances in the body which are nitrogen containing but are not proteins. That includes your nucleotides, you have nitrogenous bases in them, your phospholipids, porphyrins, and then gut bacteria, they also produce ammonia. So these are the sources of ammonia. Now, how is ammonia transported from most of the organs? Ammonia is transported from most of the organs, including brain, as glutamine. So your glutamine is the transport form of ammonia in the body for most of the organs. Okay. Why I say most of the organs? Because for muscle, the transport form is aniline. We'll just talk about it. So your glutamine is the transport form of ammonia for most organs. Then glutamine, which is formed from transamination, will accept ammonia from other sources and form glutamine. Enzyme used is glutamine synthetase. Synthetase. So if it was synthase enzymes, they do not require ATP. Synthetase, when there is a T, that is the catch point for you to always remember. They require ATP. Now this reaction is taking place in the mitochondria, and this reaction is known as the first line trapping of ammonia. So what is it trying to say? That ammonia, which is very toxic to the body, it should be care should be taken that it is not accumulated in the body, okay? Because if it accumulates in the body, it is permeable to the blood-brain barrier, so it will be able to cross the blood-brain barrier and affect your brain. So it is very important to maintain the level of ammonia. So little amount of ammonia, it can be converted to ammonium ion and be transported, but large amount of ammonia, it cannot be converted to ammonium ion because it will lead to alkalosis. So what are we doing? That we are converting this glutamate into glutamine. So how is your glutamate being formed? Via transamination reaction, you're getting glutamate. Now this glutamate, this accepts ammonia from other sources of ammonia in the body, that is from coming from your gut bacteria, from your nitrogenous substances, whatever. So, and it gets converted into glutamine. Okay, I'll just uh, draw this actually so that you're able to understand. See, your alpha amino group of amino acids, okay? They, what were they doing? They were undergoing transamination, okay? So after transamination, we've just spoken, I've explained it so many times that how they were undergoing transamination. What is being accumulated in your body is glutamate, okay? So glutamate, that is coming from your alpha amino group of amino acids. That glutamate, this glutamate will accept what? It will accept ammonia. And where is this ammonia coming from? From other sources. So this ammonia coming from other sources in the body, it is accepted by this glutamate, which is coming from this alpha amino group of amino acids to form what? Glutamine. Okay, and this is taking place in the mitochondria, and this also requires ATP, and the enzyme is glutamine synthetase. I hope synthetase. It is clear now. Okay. So your glutamine has been formed, and this glutamine, I'll repeat, I said that this is the transport form of ammonia for most of the organs. And if we see the structure of glutamate and glutamine, although I've already shown you, but still I'll recapitulate so that you're able to understand better. This was the constant part. We're talking about the variable part. So this was your glutamate. Glutamic acid, okay? It had in the side chain 2 carbons and carboxyl group. Now when it accepts ammonia, it forms glutamine. Constant part, talking about the variable part. You see, this here has been changed into amide group, so this is your glutamine. So that was about the transport of ammonia from most of the organs. And this reaction, that is your glutamate changing into glutamine, this is first line trapping of ammonia. Now moving on to transport of ammonia from muscle, because as I repeatedly said that glutamine is the transport form from most of the organs. From the muscle, the transport form of ammonia is alanine. Okay. So what is happening in the muscles? So alpha amino acid, it will undergo transamination reaction, give its amino group to alpha ketoglutarate. Alpha ketoglutarate will form what? Glutamate, and itself it will form its corresponding keto acid. Okay, so this was happening in most of the organs, but in muscle, what is happening? This glutamate will again give its amino group to pyruvate. Okay, so pyruvate will form its corresponding amino acid, that is alanine, and glutamate itself will again form alpha ketoglutarate. So this is what is happening in the muscle. So your transport form in the muscle is alanine and what? Glutamine. Now, what is happening? Your glutamate accumulates or your aniline. So your glutamate and alanine, glutamate has been actually changed into glutamine. So your glutamine and alanine, they're finally reaching where? They're reaching the liver for further metabolism. So ammonia in the form of glutamine from most of the organs and alanine from muscle comes to the liver. The glutamine and alanine are again converted to glutamate in the liver. So now in the liver, what do you have? You have accumulation of glutamate. So ammonia accumulates as glutamate in the liver and undergoes oxidative deamination. So liver glutamate is converted to alpha ketoglutarate, but unlike transamination, the ammonia is released freely here. Otherwise, what was happening? The ammonia up to now, it was being accepted by some other keto acid and it was actually accepted by alpha ketoglutarate, and what was accumulating? Glutamate. But this is the first time in the liver what is happening. The ammonia is being released freely. So I'll explain you with the help of flowchart. Okay. Yeah. See what is happening. You've received from most of the organs the transport form of ammonia is glutamine. Okay. Now this glutamine has reached liver. Now in the liver, what is happening? It is acted upon by an enzyme which is known as glutaminase. Okay. So what is happening via glutaminase enzyme? Your ammonia is released and this glutamine is changed into glutamate. So your glutamine that reached the liver for most of the organs after being acted upon by glutaminase enzyme has been changed into glutamate. Now from the muscle, you had what? You had alanine. From the muscle, you had alanine. Now this alanine, it will give away its amino group and change into pyruvate. And this amino group that was given away by alanine via transamination, it is accepted by alpha ketoglutarate. Okay. So alpha ketoglutarate will accept that amino group given by alanine to form what? To form glutamate. And this is transamination reaction. So you see, finally, what has accumulated in the liver? Glutamate has accumulated in the liver. So this is exactly what this diagram says, that your amino acids, they, after transamination reaction, they form glutamate, then the transformed form is glutamine, and ultimately again that glutamine reaches liver and it again changed back into glutamate. Your alanine from the muscle is also changed into glutamate. So ultimately the result is that you have glutamate in the liver. Now this glutamate undergoes what? It undergoes oxidative deamination here in the liver via the enzyme glutamate dehydrogenase. Now, this is oxidative deamination. You can see NAD is being changed into NADHO. There is oxidation reduction also taking place apart from just deamination. You can see this ammonia has been released freely. and this will enter urea cycle. Okay, and here you have this keto group now. So you have alpha ketoglutarate. So what was happening? Until now, your ammonia was not being released freely, but here your ammonia has been released freely. It is not. It has not been taken up by any other group. Okay, so liver has the detoxifying machinery. So how will this ammonia be detoxified? By entering the urea cycle, and then finally it will be removed. Okay. And also here that this is not a simple deamination. You can see NADH being converted into NAD being converted into NADH. So oxidation reduction reaction. It means oxidation reduction is also taking place. So that was all about your ammonia handling, until how all the nitrogen sources in your body, they've reached liver, how they've reached liver, what was the transport form, how was your ammonia handled, and now further what will happen to that ammonia which is released freely in the liver, that is, it will be detoxified by the detoxifying machinery of the liver. How? Via entering the urea cycle. So urea cycle will cover in the next class. Okay. Before ending the class, we still have 5 minutes, so I'll just give you a concept map. I'll just quickly recapitulate everything, okay? Because sometimes understanding all these metabolism and everything, it might get a little overwhelming. So I'll just recapitulate quickly in 5 minutes. Concept map for handling of nitrogen. Also talking about the toxicity of ammonia, before we begin the urea cycle, I'll also take toxicity of ammonia, that why is it so important that your ammonia needs to be handled in such a careful and regulated manner. Why cannot it just be freely there in the blood? No, it cannot, because it is very toxic for your body. Okay, so ammonia toxicity is very important. We'll take that as well in the next class. So talking about the concept map, the quick recapitulation, a summary of whatever we've studied in this class. We studied about chemistry and classification, which was very simple, only factual points. Basically, I don't think it'll ever be get asked about like in a theory question, 5 months, right, about the classification, nothing like that. Basically, you might get MCQs from that part, okay? Now talking about handling of ammonia, nitrogen, okay? So coming to that, so alpha amino acid, it gives it amino group via transamination reaction to a keto acid, which most of the time is your alpha ketoglutarate. Okay, so itself it will form what? Its corresponding alpha keto acid, and your alpha ketoglutarate will form glutamate. Then I told you how this glutamate was changed into glutamine. Which is the transport form, okay? Actually, I'll write this here. Transported as glutamine and alanine. So alanine was the transport form from the muscles and from rest of the organs. The transport form was your glutamine. So your glutamate was accepting ammonia from other sources and was changing into glutamine, and for this reaction enzyme was glutamine synthetase, and ATP was also required. Ultimately what was happening, your glutamine or alanine, it was reaching the liver. In the liver, your glutamine was again converted back to glutamate. Your alanine was also converted back to glutamate. So in the liver, what was accumulating? Glutamate accumulated. Now this glutamate via oxidative deamination, okay, and not transamination, via oxidative deamination was converted into alpha ketoglutarate. And this is where ammonia was released freely. And this ammonia which was released freely, this will enter urea cycle. Oxidative deamination. So your glutamate changed into alpha ketoglutarate via oxidative deamination, and your ammonia was released freely. And this ammonia which was released freely, it will enter urea cycle. Now, that was the concept match. So everything that we studied in this class is basically summed up in this tiny diagram, okay? So this is everything that we studied. So if you know this, you can be able to recapitulate all that we've studied. Now transamination was taking place in all the organs, okay? And now your oxidative deamination took place where? In the liver. Okay, just one last thing. Transamination reaction, which was responsible for accumulation of glutamate, is taking place in all organs. And your oxidative deamination was taking place in the liver. So both of them, they're coupled together, okay? And when you combine them together, couple them together, they are known as trans deamination. So what is trans deamination? That is your transamination reaction taking place in all the organs coupled with oxidative deamination taking place in liver is known as trans deamination. So anatomically they are taking place in different organs, but functionally they are being coupled together, okay? That is, they only have one function, that is to transport toxic ammonia. So anatomically they take place in different organs, but functionally they are coupled together. That is, their only function is to transport your toxic ammonia. Okay, so with this, we come to the end of the class. If you have any queries, you can send them in the comments. Thank you for watching.